Today we start the second video on the chapter 6, which is protein synthesis, and this is the 6.2 component of the chapter on protein synthesis. This is the second video. The first point is that a polypeptide, so a polypeptide can also be called a protein. Now what are proteins made up of? They're made up of amino acids. So we say the polypeptide or a protein is coded for. Now where do we find the recipe to make a protein molecule? And that recipe, which is present in the DNA, is called a gene. So a polypeptide is coded for by a gene. And then what is a gene? Well, basically what we have to understand, the gene is a sequence of nucleotides. Is a sequence of nucleotides. Is a sequence of nucleotides that forms part of a DNA molecule. Part of a DNA molecule. So basically you have to first understand, first of all, what is a protein. And protein is made up of amino acids. They can be proteins with 1,000 amino acids. They can be proteins with 500 amino acids. They vary. But how do we put the amino acids in what order, what sequence? Because you are only 20 different amino acids. But if there's a protein with 100 amino acids, it means that these amino acids are put in a certain sequence. But then who dictates the sequence? Well, the biggest dictator is the DNA. So the DNA dictates the sequence, and this sequence of nucleotides, remember what are nucleotides? A, T, 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 C, A, C, G, A, G, something like this, anything like this. So sequence of nucleotides. Now this sequence of nucleotides will be called a gene. Now as you can see here, this is a table which gives you the different DNA sequences. Now we say TTT, as you see this one here, codes for the amino acid PHE. But TTC also codes for PHE. So you can see this is a universal genetic code. In every organism this is going to be the same. It's not going to be any different. So as you can see here, these four all code for the same amino acid. So TCT, TCC, TCA and TCG all code for the amino acid serine. And we abbreviate it and we only use the first three letters. So what we need to understand is that this is going to be the same for every organism. Every living organism has the same universal genetic code, and it will always code for the same amino acid. It's not going to be different in different animals or different plants, but it's going to be the same story everywhere. And then you can see there are some stop codons, like this one here and this one here. Now you can see that there are 64 different possible combinations. The reason is because we have 4 bases. What 4 bases? A, T, C, G, and we have 3 bases in one for codes for one amino acid. So it's 4 raised to the power 3, and 4 raised to the power 3 is equal to 64. So 4 bases and because we have 3 bases in each set, which is going to code for one amino acid. So we have 64 possible combinations. Now in this diagram here, in the two tables, you can see we have the DNA on one side. So the DNA is TTT. So the mRNA will be AAA and the tRNA will be UUU. Now as we go along, you'll further understand this, and you need to come back to this page and then pause it and then have a look at it. And you should be able to figure out what is the DNA code, how that is going to be converted to an mRNA, and then how that is going to finally be converted into a tRNA. So you need to finish this video and then come back to this page and have a look at it again. These are the different amino acids that I am going to be talking about. So we've got this one here called Gly, then ALA, then VAL Val, then Leu, then isoleucine, which is ILE, then methionine, which is MET, phenylalanine, which is PHE, tryptophan, which is TRP, and proline, which is PRO, serine, which is SCR, threonine, THR, cysteine, CYS, tyrosine, TYR, asparagine is ASN, glutamine is GLN, and then we have aspartate, which is ASP, and then glutamate, which is GLU, and lysine, LYS, and arginine, which is ARG, and histidine, which is HIS. Now the reason why I've gone through all these is because we're going to be talking about these again and again, because in protein synthesis we have to understand that proteins are made up of amino acids, and the amino acids have to be in a certain sequence. Could be first we have serine, then we have threonine, then we have phenylalanine, then we have tryptophan. So what we need to understand is that the number and the sequence of amino acids is the primary structure of a protein. So the primary structure is dictated by the DNA. Now how the DNA results in the formation of a protein is what we need to understand. So please pause this page and just go through these names once again, and then we will proceed on to protein synthesis. Now the basic first two steps which we need to understand is that first of all, the DNA contains the gene for a certain protein. Then that gene has to be copied by the mRNA. Now this, of course, all this will going to take place in the nucleus, and that is called transcription. And I always say that you remember it like the script of a play. So this is the script to make a protein molecule. So transcription is going to take place in the nucleus of the cell in a eukaryote. And then this mRNA which is formed is going to leave the nucleus and is going to go to the cytoplasm, where translation is going to take place. Translation is what? Translation is mRNA to protein, and this is going to take place in the cytoplasm, and it is going to take place on the ribosome. So first we have transcription, which is DNA to mRNA. First it is transcription, DNA to mRNA, and then this mRNA moves out of the nuclear through the nuclear pores and then reaches the ribosomes, and then it is going to be translated into a protein molecule. So the two steps have to be understood. Number one, transcription. Number 2, translation. Transcription takes place in the nucleus. Translation takes place in the cytoplasm. Transcription, what takes place in the nucleus, the first thing we say, the gene for insulin is going to unzip, and one of this strand is going to be copied, and the strand that is copied is called the transcribed or the template strand. So this one would be the transcribed or the template strand, and who copies it is the mRNA. So as you know, in mRNA or in RNA there is U, so A will be copied by U, T will be A, then U, then U, then C, 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 G, then U again, then T, A, then U again, then U, U, A, A, G, G, C, C, U, U. Now this is not going to be that short, it's going to be a pretty long DNA which is copied by the mRNA. So we have complementary base pairing here, and the RNA has U's and the DNA, which I've shown you in black. So the DNA is copied by the RNA, but then these nucleotides have to be joined and a sugar phosphate backbone has to be formed, which is going to be done by the RNA polymerase. This is going to be an enzyme called RNA polymerase that is going to be joining these RNA nucleotides together. So the RNA polymerase is going to form the sugar phosphate backbone and join this to form a continuous chain, and then this is going to move out of the nucleus, this green strand, which is the mRNA. So this mRNA is going to now leave the nucleus and come out of the nuclear pore and go to the cytoplasm. And the DNA, the gene for insulin, is going to remain in the nucleus, so it's going to zip up again. And it's going to join together again and form the normal DNA double helix. In translation, what is going to happen? That the mRNA which came out, of the nucleus is going to go and get attached to the ribosome. So this is the ribosome, could be a fixed ribosome, could be a free ribosome, and the mRNA has got attached to it. Now whatever is written on the mRNA, let's suppose that the first 3 letters were U, U, U. Now these first 3 letters on the mRNA, the first 3, is called codon 1. And then there's another codon, so that's codon 2, and then another codon. So we have codon 1, codon 2, this is just to make you understand it, and codon 3. So UUU would be codon 1, AAA would be codon 2, and CCC would be codon 3, and 9 bases come and fit onto the ribosome. So the mRNA fits onto the ribosome, and we have understood that the mRNA contains codons. Now there's another type of molecule which is called a tRNA, and these tRNAs have anticodons on it. So if you see here UUU, then there would be AAA, which would be the complementary base pairing. Now this AAA and this blue structure is called the tRNA. And this AAA, these three bases, is called anticodon. Please remember there is a T. So these will be called anticodon. And this will always bring a specific amino acid. Like, let's for instance label these amino acids 1 to 20 with different R groups. So I'm just saying it will bring amino acid number 5. This is just to make you understand that the anticodon and the amino acids are very specific. So the next tRNAs would be UUU, and this would bring amino acid, maybe amino acid 10 or 11, any specific amino acid. Now what we need to understand is that once these two amino acids have been brought together, now they have to be joined by a peptide bond, and that is going to be joined by an enzyme, which is a very specific enzyme, which is called peptidyl transferase. So what do we have now? What we have now is two amino acids joined together by a peptide bond, and then we see a third tRNA coming over here, and the third tRNA would of course have complementary base pairing, so we have GGG here, and this could probably be bringing amino acid number 9. And then we have another peptide bond being formed. And so you can understand now what is going to happen is that more amino acids are going to be added to it. More and more amino acids are going to be added to this growing chain. And we have maybe 11 here again, and we have maybe 7 here again, and 5 here again, and then it keeps on adding new. So basically what was translation? If you remember what I said, translation was what? Translation was mRNA to a polypeptide chain or a protein molecule. To a polypeptide chain, or you can say a protein molecule. So more and more amino acids will be added to this polypeptide chain, and then there will be a stop codon, and that of course would end the protein molecule and no more amino acids would be added to it. So this is a brief video on how transcription and translation is going to take place. Your syllabus is very clear. You have to know what is RNA polymerase, messenger RNA, codons, transfer RNA, anticodons, and ribosomes. Now the syllabus for 2022 is very clear about is that we have to understand what is the transcribed or the template strand. The strand of DNA molecule that is used in transcription is called the transcribed or the template strand, the one which is going to be copied by the RNA. So this in red is the mRNA. So the mRNA is going to be copying one of the strand, and that strand is going to be called the transcribed strand. And then the RNA polymerase is going to join this. Now the other side of the DNA molecule, which is not, and the other strand which is called the non, so this one which I am coloring in blue is going to be the non-transcribed strand, because that is not copied by the mRNA, but it is there because once the mRNA leaves the nucleus, then this DNA is going to again join up together and form the complementary base pairing, and it will remain in the nucleus, and it will be of course used again whenever the protein has to be made again. Now in eukaryotes there is something known as a process which occurs eukaryotes only because they have a proper nucleus and they have certain enzymes which we need for this process, and this process is called RNA processing. Now in RNA processing what happens is the DNA contains non-coding regions. Now these non-coding regions are called introns, inside introns. They don't code for anything. We don't know why they're present. And the other areas are called exons. So we've got these introns and then we've got the exons. Now the pre-mRNA copies the whole of it, not realizing that there's a non-coding region. Now there's a special enzyme which is going to now remove these introns, which are the non-coding regions. So this part has to be removed and this part has to be removed because it is a copy of the intron. And then the pre-mRNA, this is going to come here and this is going to come here and this is going to come here. Now this is going to be the final mRNA. It's going to join together. So it's going to be shorter because we removed the introns. And this is the part which has been added new to the syllabus. Explain that in eukaryotes the RNA molecule formed following transcription, which is the primary transcript, is modified by the removal of the non-coding sequences introns and the joining together of coding sequence exons to form the mRNA. So first there is going to be DNA, then there is going to be pre-mRNA, and then there is going to be mRNA. So this is of course what we need to understand now, which is an amendment to the syllabus for 2022. Now the last point of this component of chapter 6 is gene mutation. And a gene mutation, as I have given you, is that this is the DNA. Now there is a certain sequence, and you can see I have just put little bars under it. Every 3 is going to code for 1 amino acid. Now if God forbid there is a change, now for instance let's look at a change. If there is a change here, instead of T now this has become a C. Now this of course is called substitution. Substitution means that one of the base has been substituted. So we're going to have, now when the mRNA copies this, the mRNA is going to copy it up to here, is going to be correct, but this is going to now go wrong. Because the mRNA is going to say, okay, well this is a U and this was a T previously, but now it's a C, so this is going to become a G and this is going to become a U. But previously because it was ATT, so this would have been UAU. So now what has happened is that the mRNA from UAU has become UGU. Now this is going to maybe code for a different amino acids. Sometimes the amino acid remains the same, and if it remains the same, then it's going to be called a silent mutation. But if the amino acid is changed, then that is going to result in now change in the sequence of base pairs, and this results in amino acid sequence changing. So we have an altered polypeptide. So a gene mutation, we can have a substitution. Now besides the substitution, you can also have a deletion. You can have a deletion or an insertion. Now both of these of course have more serious effects. In a deletion or an insertion you have major problems, because what happens if a deletion occurs, that means one of these bases is removed. Now you see how this is going to change the DNA format. Now before this of course everything remains the same, like AAA is fine, CCT is fine, TAA is fine, but here you had GG something else. Now it will become GGT, and this was TAT, but now it will become ATT. Now this is called a frame shift. Just like the frame of a photograph, now frame shift. So in a deletion, a frame shift takes place. Now similarly, in an insertion, we have another problem, is because what happens is that a new base is inserted somewhere here. Now in between here, we have an A. So if that is added, now you realize how this is going to change it. Now this of course remained the same before the insertion. But after the insertion, now this was GGG, but now it is going to become AGG. And this is going to shift ahead. So in deletion and insertion, both cases we have problems, because before the deletion and insertion is fine. Nothing is going to go wrong. The amino acid sequence is going to remain the same. But after the deletion or the insertion, then we're going to say frame shift is going to take place. And this will of course change the amino acid sequence after this deletion or insertion. And that could result in a very different protein structure. And this of course, if it's an enzyme, then maybe it will not function because it will become a non-functional enzyme. So this is all that we have to know about gene mutation as a result of substitution, deletion or insertion, and how each of these mutations will affect the polypeptide produced. So this finishes your 6.2 protein synthesis, and there's one more video left which will be added to this video on chapter 6. Thank you.